Chris was supposed to actually give this talk, but he got a better offer to go to the White House. So I said, okay, fine, I will cover for you. So it's a lot of the research that we're doing together, so I'm excited and honored to be able to share with you what we're looking at. First, we'd like to just address our large group and say thank you to all of these individuals who have helped with this work. You also will notice that we have quite a diverse group in terms of basic scientists and clinicians that work side by side together, really with a sincere dedication to pediatric traumatic brain injury. So what I'm going to do today is just really briefly talk about three particular approaches that we have developed in the laboratory that we're trying to then take to the clinic and implement them to the pediatric population. So many of you are probably already familiar with some of this information about traumatic brain injury, but it is an injury that is particular to pediatrics in the sense that the incidence is very high in children under 19, and it turns out it's the #1 cause of death and disability among children under 19 in the United States. So every 30 seconds, a young person is having a head injury in the United States. It does affect children and adolescents to a significant degree with a huge economic cost. And because this population is not homogeneous, we have complexity that makes it very difficult to study. So each age group, from the infants to the children, school-age children, to our adolescents, all have different mechanisms that are associated with different kinds of risks and different kinds of causes of injuries. You add on to that gender differences that really start to develop within adolescents, and this makes it a very complex problem to study. So I'm going to tell you about the first approach that we're doing, and this is work that actually Dr. Chris Giza has been working on. And his group has largely focused on trying to understand the effects of mild traumatic brain injury in these immature rats. And what they found is that even with a relatively mild injury, they found that there was a change in this NR2A subunit of the NMDA receptor. Now, the NMDA receptor is a glutamate-activated receptor. It's been termed by the popular media as the IQ gene, and its expression, or the component of its expression, is decreased around 2 and 4 days after this mild injury in the immature animal. Now, along with that, during that time period, we see a failure of these injured animals to show the appropriate increases in cognitive performance when they're environmentally enriched reared. So normally when young animals are put into an enriched environment, their cortices get thicker. We see more dendritic branching compared to the standard housed animals. And in these mild injured animals, we're seeing a failure of this kind of normal plasticity response. We've then gone to look at activation patterns, both clinically in TBI kids as well as in our laboratory animals, and show that during tasks, we see that here are control children versus TBI children. You can see the significant reduction in the activation circuitry doing a memory task. When we look at the animal models, we see the hippocampus is activated with a pharmacological stimulus as opposed to the TBI, where we really just don't see as much activation. So Chris came up with this hypothesis in regards to manipulating this glutamatergic response that we see after TBI, where acutely you get a huge glutamate release and activation, and then it's followed by this period of inactivation or depression. Now, previously, people have tried in the field to give inhibitors, NMDA receptor inhibitors, to diminish this and decrease the kind of excitotoxicity you can get. The problem is that developmentally, it ended up being a risk for younger animals because it inhibited their ability to become plastic. And so we were seeing more cell death in these models, and that did not turn out to be a very good approach for the younger brain. Now, if you give an agonist too early, you can actually exacerbate the excitotoxicity that you see early on as well. So if by giving a timed agonist stimulus during this depression phase, it's hypothesized that, in fact, we could improve the plasticity response and help the brain recover. So he went on to give D-cycloserine, which is actually an NMDA coagonist. It restored the NR2A subunits in these animals, and it actually improved their environmental enrichment. And here in the Morris water maze, we see that their latency to find the platform is actually improved as well. So this particular treatment seems to reverse some of the effects that we're seeing with the mild traumatic brain injury. And in fact, this is now currently being moved to the clinical studies with Dr. Asarno with their R21 grant to look at pre-activation and then use of D-cycloserine during a task and then post-activation of the TBI brains in children. So this study is currently underway and is an example of how we're trying to move some of these findings into the laboratory settings to help kids. The second approach that we're using is actually my area of research, which is the alternative substrates or alternative fuels. Now, normally the brain uses glucose, which is processed through the glycolytic pathway. Years of research, which I'm going to show you in one quick slide, is that we see a decrease in glucose uptake after traumatic brain injuries of all kinds. This is done by both PET imaging and 2-deoxyglucose autoradiography in the laboratories. We then see that the glucose that is taken up is shunted towards the pentose phosphate pathway to a greater extent, which occurs in conditions of oxidative stress or DNA damage. So you have more glucose being diverted. We also know that the cytosolic component of this cofactor, NAD, which is required for GAPDH to function, is also decreased in the brain. And so GAPDH is going to make it problematic for glucose to then enter the mitochondria. All of these dysfunctions or impairments or obstructions are going to decrease the ATP production from the mitochondria, making this perhaps not the best fuel type after brain injury. So we started looking at the use of alternative substrates that would enter the mitochondria downstream to all of the obstructions that we saw. So the laboratory has been looking at lactate and pyruvate in adult animal models showing improved behaviors in both the Morse-Water maze and in the alternation maze for the animals, showing improvement or recovery back to normal in the adults. Now, we're interested in children, and for me, I think the ketogenic diet is also a monocarboxylate that could be replaced. And we see here that it improves the contusion. So here we have cortical preservation when they're on the ketogenic diet. We know that the transporter for the ketone in the brain is actually upregulated in the adolescent brain relative to what we see in the adult after injuries. We've also shown that giving the ketogenic diet actually improves motor and cognitive function in our teenage rats, as well as improving energy metabolites like creatine and ATP. So there was a lot of very positive evidence that the ketogenic diet might be a good alternative substrate for the brain. And in fact, we're now in the beginning phases working with Dr. Joyce Matsumoto, who has actually got a CDI pilot grant to actually look at the phase 1 portion of this, trying to do proof of principle and administration issues of administering the ketogenic diet in the PICU. So this is another example of something that we've worked in the laboratory that we're trying to then move towards the clinical trial to help improve outcomes of kids with TBI. Now lastly, and this is really some of the most recent stuff, is pituitary damage. Now, the pituitary gland, located right in the middle of the brain, is held precariously by a very thin stalk. And when the brain moves, either in a concussion or in a traumatic brain injury of a greater magnitude, you can cause damage to the pituitary gland. Now, there's a lot of evidence out there for adult hypopituitarism after TBI and the consequences that come with that and testosterone replacement. There's a little bit of literature with children and more moderate and severe injuries. But Dr. Greco with our group, she's a postdoc with me, was particularly interested in the consequences of repeat sports-related concussions and these young athletes that are experiencing these things during adolescence. So she is actually using one of our models to look at what the effects are on the pituitary. And alarmingly, we're seeing decreased body weight, delayed puberty onset, hormone changes. We're seeing sexual function and reproductive organ changes in development. So these are causes for concern in our animal models. And so what we've done is we're starting to move this towards screening in the clinic to see how often are we seeing this, and should this be something that's a red flag for us to take notice in these adolescents who are having repeat concussions. So we're working with Dr. Kukwali to do an R01 on this pituitary damage. At the same time, we're implementing plans for screening Dr. Giza's pediatric concussion clinic patients, at least the moderate and severe cases, to see if we have enough evidence for this. And then behaviorally, we're trying to see what some of these consequences of hormone deficiencies are in the children by working with Dr. Sarno and looking at some of their cognitive functions, as well as doing imaging to look at functional connectivity as a consequence of hormone dysfunction with Dr. Neil Harris. So there's a lot going on where we're trying to implement and translate things to the pediatric condition. And this just summarizes some of those changes that we see. So hopefully we'll be able to get some treatments out there in the near future that actually offer options for some of these kids with traumatic brain injury. Thank you.
It's a great pleasure to be here, and I feel like I'm a representation of kind of a greater effort in autism here. So what I'm going to do is introduce our center to you for a second, and then talk about one very specific aspect of our research, this trying to move to modeling autism in a dish. And of course, one can't make autism in a dish, but modeling it is another issue. Let me see if I can actually work this. There we go. So at UCLA, we have the UCLA Center for Autism Research and Treatment, and it's a multidisciplinary center that involves far more than the work that I'm going to talk about today, but includes child neurologists, people from the Department of Education from different schools, med peds, neurology, and psychology, as well as linguistics. So it really is a true multidisciplinary center that really has been working, I would say, for the last 10 years to bring to bear everything that's needed to understand a complex cognitive and behavioral disorder such as autism. So the work spans genetics, molecular mechanisms, brain mapping and neuroimaging to look at the brain circuits, behavioral and pharmacologic treatment. And we have an unprecedented 3 Autism Center of Excellence awards from NIH out of the 12 that are awarded nationally. And this really reflects the excellence in the Department of Education and Semel Institute, as well as our other collaborators here. So we're very excited about the ability to be here today and to talk with the greater pediatrics community as well, because we have great interest in doing larger scale collaborative work and to raise raise all the boats in the campus really around this area. So we're very outward looking. So essentially, this is the model that I work with as somebody who is starting from the point of human genetics. It's kind of, identified genes that cause the disorder, gives you a clear kind of causal anchor. Then you can move from there. You can study the disorder in human patients, in mouse models, actually genetically engineered mice. We can even look at human postmortem tissue, although that's been challenging because there's very little available in the world. In fact, right now in our laboratory, we have 113, we have brain samples from about 800 different samples that are 10 different regions from 113 patients, and that really represents about 80% of the kind of cases available in the world. So even that's a relatively small amount compared to, let's say, Alzheimer's disease or schizophrenia. And of course, what I'm going to talk about today is the use of human neural progenitors. And now what I show on the left is the kind of typical way of thinking about things, the finding genes, modeling, this kind of circle, and it is a cycle that the modeling in animals gives us mechanistic insights that then we go back to patients, and patients inform the modeling, et cetera. But our real goal is on the right, to have a more integrative systems biology approach. And that's essentially what we've been trying to do. I can't say that we're there yet, but we're relatively close of integrating what is called phenotype data, that is the clinical information, with gene expression, epigenetics, copy numbers, sequence data, et cetera, multiple levels of omic type evidence. And that's essentially, now, one of the reasons we can do that is because autism genetics has identified dozens and dozens of causal genes. But first, I'll just give you a brief introduction to kind of how one way to look at autism. Many of you know, especially the clinicians in the audience, that autism is seen and overlaps with many other neurodevelopmental disorders. At least 10% of kids with autism have seizures or epilepsy in very particular developmental epochs. About 30% of kids with autism have intellectual disability. So one of the core questions is, how does autism differ from intellectual disability? Clearly, 70% of the kids with autism don't have intellectual disability, so that there are parts that overlap and parts that don't. And that's another key component that we've been working on that I won't have time to discuss today, but we published a little bit on that. So the real challenge is that autism is really conceived of fundamentally as a disorder of social cognition and behavior. And with it, there occurs restrictive and repetitive behaviors. And previously in DSM-4, language delay and language disorder was really part of the really universe of autism. And now in the last incarnation in DSM-5, that's changed. And so one of our approaches has not been necessarily to kind of consider autism as a diagnosis, as a category, but to look at the dimensions of language, of social cognition, of repetitive and motor behaviors, et cetera, within this population. And so, one of the points that I want to make is shown there on the left is an ideogram from 2008, where the human chromosomes are colored in. You can see that basically every chromosome has an autism locus on it. So right now, our predictions from genome and exome sequencing are, there are many genes, probably right around 1,000 at least, that harbor de novo rare variants. There are many genetic models. It's not Mendelian necessarily. It's not complex. There's a whole range. There are recessive forms of autism, dominant, X-linked, as well as multigenic. No major gene that's been identified accounts for more than 1%. So if you have 100 children with autism, you have 100 different genetic etaologies. Yet despite that, in autism postmortem brain, we have identified at least that 60 to 70% share a molecular pathology. So we think that there is some convergence in brain down the road. So this kind of, way of breaking up, way that I approach, research is that in a disorder like that, we need to have human relevance. So we study postmortem human tissue, and we do imaging and live human studies. But the difficulty is it's very difficult to develop a drug doing that. You need higher throughput methods that are human relevant. Of course, there's the mouse putting the human on the wheel. We can do these live human studies, but of course they're limited. And what we can do, we can take a mutation that causes autism in humans and put it in a mouse. But again, for screening, we really need to have a higher throughput system, and that's where what I'm going to talk about comes in the last 5 minutes. So the first thing we've been trying to do is develop a system in a dish using primary neural progenitors from human brain. And so we take donor tissues, and this project is the brainchild and has been run by these two extremely accomplished postdoctoral fellows in the laboratory. So what we do is we develop them, and we've been trying to validate first before, our first issue is this a valid system? And so we look at it, the neurons morphologically in a dish, and we can do transcriptomics, measuring all the RNA expressed, and we can ask, how does this look like human brain? And so this is just showing some of the morphology and the kinds of things one can do. And this is the kind of gold standard in the field, and I don't think it's sufficient, because you can only look at a few proteins at a time with antibodies. But these are we can get beautiful neurons in a dish. That looks like a beautiful neuron. At least I think it's beautiful. There are filopodia and some spines, and so we can get synapses. We can get development in a dish. But the critical question is, how much does this look like a normal human brain? And how much does this mirror human brain development? So we were involved in this BrainSpan project with folks at Yale and USC and the Allen Institute, where these 15 stages, 1 through 7 being embryonic, and you can see late fetal being period 7, where these brain regions from all of these stages were profiled using a technique called RNA sequencing that allows us to look at all the genes expressed. And so we have now an atlas of brain development that can serve as a standard. And so we also have a laser capture microdissection that was just published from human fetal brain with very high spatial resolution. So this provides our template. So in doing this, we did 3 things, and I'm only going to talk about one because of time. We first asked, how do the genes go up and down using a hypergeometric test that we call context? In In other words, how does it match, the genes that go up and down match in vivo and in vitro? And the second, we developed a machine learning tool that actually allows you to take a culture, a single culture that you've developed, and ask what part of the brain does that come from and what stage is it most representative of? And that's really going to be very critical and very useful. And we validated it on multiple data sets in vivo and in vitro. But what I'm going to show you right here is now using network biology, where we can look at how genes are co-expressed in neighborhoods and ask, how do biological processes, how are they conserved in vitro? And I'm just showing a few here. This is a timeline showing the cortical development and the stages, and basically neuronal birth is occurring here. And then this is the time before birth where there's maturation, programmed cell death, dendritic growth, synaptogenesis, and then the onset of gliogenesis. And you can see these different colored, these magenta is a group of genes. It's a module of highly co-expressed genes that share the same neighborhood involved in this process. And essentially, what I'm showing here is that these processes are highly conserved. This is just showing some examples. This is related to the inhibition of neuronal development. And you can see it's high early, and then when neurons start being born and migrating, it goes down. And then when glia start being born, it goes up again. This is another module that's related to proliferation, again, going down and staying down. These are synaptic modules and various aspects of synaptic development, etc. We can also ask, how are all these processes, and they're grouped into modules that get random color codes that just arbitrarily assigned. So each of this corresponds to a group of several hundred genes that are co-expressed during brain development. And we can ask, how do autism or schizophrenia or Alzheimer's genes fall into these? One thing you can see is that genes that cause late onset diseases aren't expressed at high levels together in these cells, as you might expect. But a number of the autism genes from autism brain, genes that we and others have discovered as mutated in probands, are strongly enriched in this one particular midnight blue module, etc. So we're able to ask and show that there's conservation of particular processes of human brain development in a dish. We're able to do that rigorously and quantitatively. And then we're able to ask, are these genes or processes conserved? And many of them are, and they allow us to now start to model. So we have shown that we have brains that go from about stage 1 to about stage 6 in a dish, that these processes are conserved, that they have translational potential. And work that I didn't show shows that these primary neuroprogenitors preserve in vivo development slightly better than many of the other models out there right now. And so we still have a ways to go in some of them. So we have a website where you can now take your data from an in vitro system and ask how well does it match in vivo development. So the project is essentially, now that we have this validated system, we can put autism risk mutations, we can follow them over development in a dish, study morphology, physiology, function, as well as the transcriptional signature at a genome-wide level. We can use that transcriptional signature to search for drugs that will reverse it in various databases, such as the connectivity map, and then attempt to rescue the genetic mutation with drugs. And that's what we're doing right now. We're also asking, if we have 10 or 20 different mutations, do they actually converge on common pathways at a transcriptional level? So these are the folks I have to thank. Again, I mentioned Luis and Jason, who are running this project. Our funding sources here, including the Simons Foundation, the NIMH ACE Centers, the Broad Stem Cell Research Center, which supports Luis, and we have an innovation award for some of this work. The other folks in the laboratory who are involved in network analysis, many shown here, as well as work that we've been doing with the Gage and Moitri laboratories at UCSD and Salk, and the Cossack laboratory at UCSB to try to improve the modeling of iPSC-derived models based on these transcriptomic signatures. Thank you.